For our research presentation, we did the article, The Role of Subcortical Structures in Human Epilepsy.
Theoretical framework. Seizures can often occur during changes of brain activity and can be caused by a variety of factors. Instead of focusing on simply where the seizure starts, the study focuses on how seizures are not just localized to one region. The study utilizes a network neuroscience lens that investigates how numerous different brain regions communicate through epileptic seizures and how their functions are influenced. Through both human and animal studies of epilepsy, the question of subcortical involvement in epilepsy provide framework for how seizure behavior, such as loss of consciousness, happens to develop. Research questions and hypothesis. The overall research question in this article states, what roles do subcortical structures such as the hypothalamus, thalamus, cerebellum, and brainstem play in starting, spreading, and causing symptoms in humans experiencing epileptic seizures? The hypothesis states, seizures that start in the cortex or outer layer of the brain directly impact subcortical structures of the brain, which in turn disrupts and slows down other subcortical areas that are not seizing. This would ultimately explain why individuals sometimes lose consciousness during epileptic episodes.
During this study, the subjects and models were cats and monkeys with motor cortex-related seizures, guinea pigs with chemically induced seizures, rats with experimentally induced motor cortical epilepsy, and 4 humans with uncontrollable focal seizures. What was manipulated? There were lesions or alterations that were made to different thalamic nuclei, the VA, which is the ventral anterior, VL, the ventrolateral, and VPL, the ventral postolateral. The few different outcomes that occurred in animals, lesions in the VL, VA, or VPL reduced seizure frequency, lowering the VL temperature resulted in a temporary block of seizure activity, stimulating the VA increased seizures, and lesions in the posterior nucleus resulted in a change in brain metabolism during seizures, but did not stop the seizures. And in humans, the VA lesions in 4 patients with drug-resistant seizures, 3 patients experienced an initial decrease in seizure frequency, but it did not last. 2 of these 3 patients showed fewer generalized seizures after the VA lesions, and the 4th patient showed no improvement at all.
Findings and what they mean. The authors found evidence that subcortical structures and cortical structures both have an important role in seizure activity. They found ample evidence suggesting that substructures within the diencephalon, basal ganglia, cerebellum, and the brainstem are all involved in the pathophysiology of human epilepsy. To be specific, the thalamus and brainstem most likely regulate the cortical excitability and the seizure threshold. They found subcortical structures have been seen to represent the start of origin for seizure activity. What this means is that the subcortical structures should not be overlooked because of the important role they play. Some real-world implications is that by paying attention and looking more closely at these subcortical structures, we can find some potential therapeutic targets for seizures.
Our questions for the class include, when you think of seizures, do you automatically associate them with one particular area of the brain? Why do you think it's important to study both animals and humans when it comes to epilepsy research? What theories might you have regarding why individuals lose consciousness during seizing?